Hello everyone, my name is Sandeep Patil and today we are discuss on the drug discovery to the regulatory bodies. So starting from the drug discovery means to it is a process throughout the potential medicines are identified. That means you develop a that means you develop a new drug. To discover a new drug. So uh, basically it's contain four stages where they are as follows: target identification, target validation, lead identification, and lead optimization. Where the target identification, where you identify the disease; target validation, where you identify the organ the disease mostly affected to the particular organ. Lead identification, where you identify the compound that can be optimized into the drug. And the lead optimization, that means the lead compound will be optimized into a required drug. When there are the different forms when you apply for to NDA, just like a treatment NDA. Treatment NDA means treatment NDA. It's mean take a approval of drug which is still in a clinical phase, but patient need it for the treatment. It is the emergency protocol. The hospitals send this approval to the company, and company take that treatment IND form approval from the FDA, where the SNDA form is also available for a supplemental new drug application, where any new things you want to add it, you have to fill this form. You have to complete this form. Abbreviated new drug application, where you want to get approval for the production of generic drug. There are different types of drugs, just like a orphan drug, where this is the rare drug used in a rare disease. Another is a prodrug. Prodrug is a what do you say? Prodrug is a which have no side effects and will have a 100% bioavailability with the affordable sources. Affordable sources. That means a gold drug. Lead compound is a chemical or natural entity will be optimized into a drug. Another is a prodrug. Prodrug, it is a entity which will be optimized after the metabolized into the body. So there are the four types of phases. Phase 0 is five types of phases. Phase 0 is the microdosing. Phase 1 is a healthy volunteers. Phase 2 is a therapeutic confirmatory trial and exploratory trials, and the phase 3 is a therapeutic confirmatory trial, and the fourth one is a post marketing service. Second one is phases of clinical trial. Main purpose of clinical trials, that means clinical trials is a systematic investigation on a human subject to Evaluating the safety and efficacy of new drug. Phase 0, these are the five types, mainly four types of phases. They starting from the phase 0, that is microdosing, where the 0 to 100 mg of dose given to the patient and check the changes made in the patient. Phase 1, uh, another is phase 1. Phase 1 is a healthy volunteers, when 20 to 25 healthy volunteers are taken, and minor quantity of dose taken to that patient, and check the uh, check check the frequency of the healthy volunteers. Whereas the volunteer does not have any disease, just like AIDS, or they don't have taken any drug therapy. This phase is designed to understand the safety, tolerability, pharmacokinetics, and pharmacodynamics. It contains a SAD, MAD, and a food effects, where the SAD is a single ascending dose studies, and the MAD is multiple ascending dose studies, and food therapy is a investigation different foods any cause effect on a drug. Phase 2 is a uh, therapeutic confirmatory. Phase second is a therapeutic exploratory trials, where the 200 to 300 patients are taken. Um, this phase is mainly designed to understand the safety, efficacy, and side effects of the drug. Phase 3 is important because it's want a long period for the performance, where it is performed on a 100 to 3,000 patients. It may take up to 5 years of time to establish the efficacy of that drug. It's mainly designed to understand all the things: pharmacokinetics, pharmacodynamics, safety, side effects of that particular drug. Phase is the fourth one is the post-marketing surveillance. After getting approval from the NDA, the drugs introduced in the market, and then continuously monitoring to the drugs. They get the information of that drug, uh, how it's affect on the patient, or any types of side effects it shows. Where uh, they get this all the information from the hospital, um, MR, or the what do you say, pharmacist, um, and continuously monitoring by the PV, where the PV is working. Third one is a pharmacovigilance, where according to WHO, pharmacovigilance is a science and activity related to the detection, assessment, collection, understanding, and prevention of any side effect of the drug, or any problem related to the drug. So, in a simple words, pharmacon means drugs, and vigilance means to keep watch. That's entire term means to continuously keep watching on drugs. Drug, uh, where the four parameters are very important in a PV: detection, assessment, understanding, and prevention. Where the detection, the investigator or the healthcare professional, sorry, the investigators are responsible for the to detect any ADR, side effect of a drug assessment where the investigator or healthcare professional will be assigning the adverse event or any other drug related problem. To understanding the reporter and safety specialist is involved to understand the adverse drug events and prevention to take proper action when it needed to stop that adverse effect or communicate with other regulatory authorities so it will not happen in the future. Aim and scope of PV is a patient care, public health, risk benefit assessment, communications. The objective of PV main objective of PV is to reduce the frequency and severity of adverse event, and the another one is if you want the quality of product, you have to you have to check all the clinical trials and manufacture. Basically, the quality of product depends on the pharmaceutical industry. When you give them a approval, so you need to check all the parameters of the new drug. PV is introduced in India in 1992 and 1989. ADR monitoring system for India. In 1997, India joined with the USA ADR monitoring. 2004, the National PV program are introduced, and finally in 2010, Pharmacovigilance program of India PVPI started. It is headquartered in Ghaziabad. Ghaziabad. So all the PV are concerned with ICR individual case study reports where the different software are used for the repeating these ICRs. ICR's overall review is like first of all receive the case, then validation of duplicate case in which they are checked, the double cases are registered, then logging of case, then triage that's mean they study the case are serious or non-serious, then they enter data entry, quality review, medical review, then close the case and generate the data for the submissions. Regulatory bodies, last one is regulatory bodies regulate the guidelines through the monitorings. They are basically three primary functions: the regulation, guidelines, and compliance. Where they regulate, create the guidelines. If you want to approve, you have to work according to guidelines. There are different regulatory authorities in India: CDCSO, Central Drug Standard Control Organization; USA Food and Drug Administration; UK is Medicines and Healthcare Product Agency; Japan has a Ministry of Healthcare and Welfare; Canada has Healthcare Canada; Australia have a TGA, Therapeutic Good Administration; and Europe have a European medicines. There are different reporting forms. Just India have a HDRRF, suspect adverse drug reaction reporting system. UK has a yellow card. US have a MedWatch, and Europe have a COMS. Basically, the Argus is a software where are used for the reporting. That's it. Thank you. In that particular case, software used to reporting ADR through the ICR is India have a Sugam, UK has a Farm Ready, sorry, US have a farm ready, UK has a MHRA vigilance, and Europe have a EV web. That's it. Thank you. This is overall summary of drug discovery to the regulatory authorities.